In today's episode of Shareable Science Beyond the Blog, let's explore monoclonal antibodies, what they are, how we identify them, and why they could be a key tool in both the treatment and maybe even the prevention of COVID-19. Welcome to Shareable Science. Science you can share. This week brought news of the first clinical trial using antibodies as a potential treatment for COVID-19. We've talked about antibodies on other episodes of Shareable Science Beyond the Blog, but let's spend some time exploring monoclonal antibodies in more detail. To start, I'm going to use an analogy. Now, let's imagine that this is my closet. Now, some of you who have spent much of the pandemic in your pajamas may not even remember what's in your closet. But for me, my closet's made up of lots of different clothes. Some clothes that are more business appropriate, more formal, some that are more casual and comfortable. And every morning I decide the specific combinations that I'm going to wear in order to be about my business that day. So there are lots of different things that I could choose. I could choose something if I'm making a presentation. I could choose something if I'm feeling more outdoorsy. I might choose something for an afternoon outside on a warm summer day. Or maybe something just hanging out watching Netflix. I might even choose some combinations that don't look as well as I might have originally thought. So, fashion do's and maybe some fashion don'ts. But the concept is every day I choose the individual articles of clothing that then make up the final outfit for how I'm going to present myself through the day. All right, there's the analogy. So I want you to hold with that closet analogy. Now, let's go to the science. We're going to talk specifically about a type of immune cells called B cells. And these B cells become, or one of the things that they become, are plasma cells. Now, heads up, we're going to do a lot of immunology in a really quick window of time. And I'm going to talk really, really fast, and we're going to move, no. We'll walk through this at a regular speed. But these plasma cells produce antibodies. They are antibody factories. They produce thousands of antibodies per second. And each plasma cell produces one type of antibody. For COVID-19 therapy, for lots of other therapies, you are looking for that specific antibody that, in this instance, binds to the SARS-CoV-2 virus and blocks it from entering into a human cell. So there are lots of different antibodies, some that don't bind the virus at all, some that bind the virus but don't block it. We want that magic combination of I recognize this as the virus, I bind it, and I block it. So those are called neutralizing antibodies. Now, the body is an amazing organism. You and I, our immune systems are incredible. We generate so many different types of antibodies for all different kinds of needs. Here's how that works. We're going to talk about some genetics for a second. So, in every single one of these B cells, when they are early in development in your bone marrow, they have a whole option of genes that they can choose from to help build what ultimately becomes the antibody that they're going to produce. So we're now back to the analogy of the closet. Lots of different choices. And each B cell picks one of each category and puts that together, and then that is the specific recipe that helps make the antibody for that B cell. So two different B cells can choose very different combinations, just like I could choose very different sets of clothing to make different antibodies. There are literally billions of possible combinations for antibody production. Some of those are the equivalent of fashion fails. They do not do what we want, and those often get weeded out. But often you will find that right combo that makes this neutralizing antibody. So with that in mind, our goal for trying to find a treatment or a preventative for COVID-19 using antibodies is to find the right combination that gives me the antibody that does the job I need. So here's the process in a nutshell. How do I identify which antibodies are best? And that generally comes from plasma, from blood, from patients who have recovered, who already are making antibodies. Their immune system is doing what it needs to do. And so now a scientist looks at all those different antibodies and tries to identify the right candidate, the best candidate that binds, that neutralizes, and importantly, does it at low concentrations. Because if we're going to make this in the lab and give this to millions and millions of people, we need an antibody that's incredibly potent, that works at low doses. And then you have to figure out what gene combination, back to the closet analogy, which specific combination of genes actually gave you this fantastic antibody. And then how do we manufacture that in a lab in large enough doses that we can actually run clinical trials with patients to say, does this provide a treatment? Does this help speed recovery or minimize the severity of symptoms? Or maybe even, if I give this to people ahead of time, would it give them some short-term immunity and protect them from getting COVID-19? And then once you've gone through that process and you've found your ideal antibody, or maybe different companies look at different antibodies, then how do you mass produce this to give this to millions and millions of people around the world? We've moved through a lot of this and continue to move through this incredibly quickly, and we're at our first clinical trial. You can get into more detail about this with this fantastic reference from the Scientist magazine from June 2nd. First antibody trial launched in COVID-19 patients. It goes into this in much more detail, and if you find this fascinating, you will enjoy that article. If you found this useful, please share this with those around you. Thank you for watching. I look forward to seeing you again next time. Take care.